Students, this video is based on your chapter Respiration in Plants. So we know that all the organisms need energy for their life activities, and oxidation of food results energy production. So we can say that all the organisms need energy to proceed their vital activities. And this energy will be provided by the oxidation of food. And two main physiological processes are involved to produce energy. One is digestion and another is respiration. In case of digestion, the large macromolecules are broken into smaller macro, macromolecules or micromolecules, and these macro or micromolecules will be used in respiration, and in presence of oxygen the energy will be produced in the form of ATP, adenosine triphosphate. And this, the whole process of oxidation of food in the cell is called as cellular respiration. The breakage of carbon to carbon bonds of complex molecules, or the molecule respiratory molecule through oxidation within the cell leads to release of energy. This is called as respiration. Means what happens? The bonds will be broken, and by the breaking of bonds, the energy will be released. And this release of energy will be called as respiration, or more specifically we can say cellular respiration. Usually organisms utilize carbohydrates for the oxidation and to get the energy. But sometimes, and in some organisms, proteins, fat, and organic acids are also used as the substrate to oxidize to produce the energy. The gaseous exchange takes place in the plants through lenticel and stomata. So we know that the higher plants or the higher organisms you oxidize their food in presence of oxygen. So the essential requirement for the oxidation of food is oxygen. So we will see some modifications in the plant, how the plant exchanges gases and how they get the oxygen. So the gaseous exchange takes place in plant through lenticel or stomata. Means for the oxidation of food, the plant cells will receive the oxygen through lenticel and stomata. They also utilize their own oxygen as a process of photosynthesis, as we know that a huge amount of oxygen will be released. So that oxygen will be utilized by the plant cells itself and will be used for the oxidation of food, and they do not need much oxygen because their rate of respiration is very slow as compared to animals. Why the rate of respiration, and why they need very less amount of oxygen? Just because they have very few cells in living condition. Means they know that most of the plant cells are dead, and some of the plants are present in the living condition. So those uh, those living cells of plants are present near the surface of the plant as to receive the oxygen easily and efficiently. And these cells are present beneath the bark as to get the oxygen directly through lenticel or stomata from the environmental gases, and they are loosely packed. Means the living cells present in the plant is loosely packed. So the loosely packed cells will provide some air spaces. Through that space, the air or oxygen will move. Now we can see the general formula of glucose combustion and the combustion process, we can see here that glucose has been oxidized and has been broken into carbon dioxide and water, and ATP has been produced, and this ATP is known as energy coin. It means we are living organisms utilize this ATP molecule to receive the energy. Whenever when the organism will need the energy, then this ATP will be broken into ADP, and ADP will again can be broken into AMP, adenosine monophosphate, and the breakage of bond energy will be released. So some of there is one fact that some cells and some organisms do not utilize oxygen for the respiration process. It means this is not a thumb rule that all the living organisms utilize oxygen for the oxidation of food or glucose. Some of the animals are or plants and some uh, microorganisms are also there. They utilize carbon dioxide, or the process will be proceeded in absence of oxygen for the oxidation of food, and energy will be liberated. Now, the breakdown of glucose is a step-by-step process, and the first set of reaction, including breakdown of glucose into pyruvic acid, is called as glycolysis. Now, we will go in detail of this glycolysis pathway. So, glycolysis takes place in cytoplasm, and this is a common pathway in aerobic and anaerobic respiration. It means in aerobic respiration and in anaerobic respiration, glycolysis pathways operate. Means the glycolysis pathway doesn't need oxygen. Means it is oxygen independent process, and we can understand the uh, meaning of glycolysis is glycose means sugar and lysis means splitting. Here, the splitting of glucose will be done. The pathway was first described by Imden, Meyerhoff, and Farner. So that's why this pathway is also called as EMP pathway. In this process, glucose, which is 6 carbon molecule, undergoes partial oxidation, and it forms at the end of the pathway into two pyruvic acid molecules. It means 6 carbon glucose molecules will be broken into 2 pyruvic acid molecules. Before starting the pathway, as we know that plants store the food in the form of sucrose. So sucrose is converted into glucose and fructose by the action of an enzyme invertase. We can see here the sucrose has been converted into glucose and fructose. So these glucose and fructose will go in the glycolysis pathway and will produce two molecules of pyruvic acid. And it is a uh, uh, in a if we want to describe this glycolysis in a definition form, then we can say that it is a chain of 10 reaction process under the control of different enzymes takes place to produce two pyruvic acid molecule, and it is of 3 carbon molecule from one glucose molecule, and it is 6 carbon molecule. This process utilizes and synthesizes ATP or NADPH plus H plus. It means this process is all about to convert glucose into two pyruvate molecule, and utilization and synthesis of ATP and NADPH plus H plus occurs. Now we'll go in detail of the glycolysis process. We can see here that this glucose has been converted into glucose 6-phosphate by utilization of an ATP. Now this glucose 6-phosphate is converted into fructose 6-phosphate by action of an enzyme phosphoglucose isomerase. Now we can see here that fructose 6-phosphate has been converted into fructose 1,6-bisphosphate by utilization of an ATP. Now this fructose 1,6-bisphosphate has been converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now we can see that starting from glucose to fructose 1,6-bisphosphate, that each and every molecule is of 6-carbon compound. But when this aldolase enzyme works, then what happens? This fructose 1,6-bisphosphate will be broken into two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these two molecules are interconvertible. Now we can see that this glyceraldehyde 3-phosphate has now been converted into 1,3-bis phosphoglyceric acid by the generation of NADH plus H plus. Now this 1,3-bis phosphoglyceric acid has been converted into 3-phosphoglyceric acid by the generation of an ATP, and this 3-phosphoglyceric acid has been converted into 2-phosphoglycerate by the action of an mutase enzyme, and this 2-phosphoglycerate has been converted into phosphoenol pyruvate, and by the action of this enolase enzyme, water molecule has been released. Now this phosphoenol pyruvate has been converted finally into the pyruvic acid, and one ATP has been generated. So we will see here that how many ATPs or NADPH plus H plus has been utilized and released. So we can see here in this process, the first process here, from glucose to glucose 6-phosphate, one ATP has been utilized. Here in this process, from fructose 6-phosphate to fructose 1,6-bisphosphate, again one ATP has been utilized. So we can see here in this process, two ATP has been utilized. Now we can see here that from glyceraldehyde 3-phosphate to 1,3-bisphospho phosphoglyceric acid to NADPH plus H plus has been generated. Why two molecule has been generated? We can see here two molecules of this glyceraldehyde 3-phosphate has been formed from one glucose molecule. That's why the two molecules will be generated here. Again, we can see here in this step 1,3-bis phosphoglyceric acid and 3-phosphoglyceric acid. We can see here uh, in one reaction, one ATP has been generated. Since two molecules are there, that's why two ATP has been generated. Now we can see here in the last step, from phosphoenol pyruvate to pyruvic acid, one ATP will be generated in one reaction, and since two phosphoenol pyruvates are there, that's why two ATP will be generated. So we will see how many total of ATP has been generated. So only if we will talk about ATP, then a total of 4 ATP has been generated. Now we can see how many NADPH plus H plus has been generated. So 2 NADPH plus H plus has been generated, and this reaction process consumes 2 ATP also. It means if we will talk about the net gain of ATP, then 4 ATP minus 2 ATP a total of 2 ATP has been generated. Now, this NADPH plus H plus will be converted in some pathways, and it will ultimately form 6 2 2 NADPH plus H plus will form 6 ATP. So we can see here that 6 plus the two rest of 2 ATP, a total of 8 ATPs will be gained by this glycolysis pathway. And if we will talk about the ATP only, then the net gain is 2 ATP. So this pyruvic acid produced can be handled by different cells in different ways. It means since this pathway takes place in aerobic and anaerobic both kind of respiration, that's why this end molecule, the pyruvic acid, will be handled by different uh, cells and in different conditions in different ways. So first we will talk about fermentation here, but before starting the fermentation, we will see some glimpse about the fate of this pyruvic acid. So this pyruvic acid can be utilized by fermentation, means can be utilized in fermentation process by some organisms, and this pyruvic acid can be utilized in aerobic respiration by some organisms. So in case of fermentation process, it can be formed, uh, this pyruvic acid can be converted into lactic acid or alcohol. So one fermentation process is lactic acid formation, in which the pyruvic acid will be finally converted into lactic acid by the process of anaerobic respiration, and in some organisms this pyruvic acid will be converted into alcohol, and again this is an anaerobic condition, and in we can see here that pyruvic acid has not been completely oxidized, means we can see here the end product is either lactic acid or alcohol. It means incomplete oxidation takes place in case of fermentation process. But on the other hand, we can see here that aerobic respiration, in aerobic respiration, the complete oxidation of glucose takes place as to form carbon dioxide and water. Means after glycolysis, two pyruvic acid molecules will be formed, and this pyruvic acid will be utilized in Krebs cycle, and in presence of oxygen, the complete oxidation of this pyruvic acid will be done, and it will be broken into carbon dioxide and water, and energy, a huge amount of energy will be released. So we can see here the fermentation process. So in case of fermentation, as I have already told, that glucose will be converted into pyruvic acid. We can see here 4 ATP will be utilized, 4 ATP will be generated, and 2 ATP will be utilized. Means out of total 4, only 4 will be remain. Out of 2 ATP will be remain, and 2 NADH plus H plus will also be generated by the process of glycolysis. Now we can see here that this pyruvic acid, when will be utilized by some bacteria and in muscle cells of animals in absence of oxygen, this pyruvic acid will be converted into lactic acid. It means some bacteria and in muscle cells of animals in absence of oxygen, this pyruvic acid will be converted into lactic acid. And we can see here one molecule of NADH plus H plus has been utilized. And in case of yeast, this pyruvic acid will go through uh, ethanol fermentation process, and finally ethanol and carbon dioxide will be produced, and one molecule of NADH plus H plus will be utilized. So we can see here the fate of glucose in case of fermentation process is either lactic acid or ethanol. And we can see here that not much energy has been released in this case of fermentation process. We can see here that this NADH has been utilized here, and 2 ATP has already been utilized. So very less amount of energy will be utilized. Now we can see that sometimes it became hazardous either of lactic acid or alcohol formation. Means this fermentation process can be hazardous to that particular organism. As for an example, when the alcohol reaches about 13% or more, then it leads to death of yeast. Thank you.